traditional owners of the land on which we live, the Camaragal people of the Eora Nation. It is upon their ancestral land that Winona and its surroundings stand today. I pay my respects to the elders past, present, and those of the future, as they are the people we remember, respect, and represent to carry on Indigenous custodianship and care of the country.
Welcome, people who are here in the audience with us and those joining us here with the live stream. Tonight we are here to celebrate our 21 marvellous students. Although they are not all here tonight, we do have them speaking to us via video, so they are all represented. We are going to show you some short video segments of their work, and then we will have an opportunity to ask them questions afterwards. So I would like to invite our first group of speakers up to the stage and welcome you to their video.
We reference to biopesticides on a significant test in the agricultural industry, the helicobacter, and I basically found that while both biopesticides were effective, one was genetically modified and one was a virus-based product, so it was extremely hard to compare those in a field test, so I wasn't able to directly compare them, but I found that both of them were extremely effective in killing and raising the mortality rate of helicobacter moths. And I found this research really important because chemical pesticides are a significant issue in the Australian agriculture industry. They cause a lot of runoff, the Great Barrier Reef is suffering from it, and I think a switch to an ecological alternative is really important, so I thought that's where my research would go.
So I researched into the effect of Barbavax vaccine in reducing the worm egg count in relation to Haemophilus contortus. Haemophilus contortus is a highly pathogenic nematode that is causing billion-dollar losses to the agriculture industry. And the effect of in reducing the worm egg count was to a limited extent, as the Barbavax vaccine has to be constantly used and doesn't allow for just one dose and then complete resistance to Haemophilus contortus.
I've been researching the effectiveness of the COVID-19 vaccine versus unvaccinated. I chose this topic to educate people about what COVID-19 is and provide knowledge and scientific background behind the virus. I also chose it to educate people through data to get vaccinated by providing the potential long-term effects COVID has on unvaccinated and vaccinated. My hypothesis was that getting vaccinated against COVID-19 is better for your immune system than staying unvaccinated. The dataset reveals a stark difference in infection rates between the unvaccinated and vaccinated populations, with the latter showing significantly lower rates of COVID-19 infections, and furthermore, the analysis highlights that individuals above the age of 50, especially those with pre-existing health conditions, benefit from vaccination as it neutralizes the body's reaction to the virus, reducing the risk of severe outcomes.
After hearing about zombie diseases online and in the media, I decided to research about how they spread. Called zombie diseases because of their symptoms and fatality, prion diseases are incurable and are spread when a host ingests infectious prions from an already diseased animal. I wanted to understand, is there a relationship between climate and environmental factors such as temperature, precipitation, sea level and topography, and the transmission of prion diseases? This research is vital as it can lead to a decrease in the number of infections and fatalities, and reduce the detrimental effects on human and animal welfare that these diseases have. I hypothesized that changes in climate and environmental conditions significantly impact the transmission dynamics of prion diseases. My results found a statistical connection between reduced temperatures to increased CWD cases, which is a type of prion disease. I also found a correlation between increased water bodies such as rivers in areas of increased cases.
We'd like to open up. Does anyone have any questions they'd like to ask these girls about their projects? I'm sure they're dying to have unexpected questions. Oops.
Here's my teacher voice. They're such fascinating topics. I wonder what was the inciting incident that made you decide this is where to direct my project. What was your aha moment?
because you don't have to pick the science that you do. Like I do urban environmental science, and there's only like 4 people in my class. But I love our science, but I wanted to do something a bit different because I like a lot of different areas. And I don't know, I just looked up interesting things in science at the moment, and I saw zombie diseases. And that was interesting.
So for me, I live on a farm, so we have livestock, of course, and I've kind of been surrounded by that growing up. In relation to Haemochus contortus, or Barber's poleworm, I just thought it sounded smart, Haemochus contortus, but Barber's poleworm, it isn't on my farm, but it is causing billion-dollar losses to the industry. So that's why I kind of researched into that, and I wanted to do sheep because that is on my farm as well.
Mine's kind of a parallel with Lucy's, but I also grew up on a farm, and when I took science extension, I kind of went to my dad and said, I asked if there was any issues that he faced, and he said Helicobacter moss, particularly recently, he'd had an issue with in chickpea and canola crops, so I decided to research that, and the sort of ecological alternative to pesticides, to chemical pesticides, which can cause a lot of issues with not only the humans handling them, but also sort of interrupting the ecosystem dynamics. So I was really interested in finding out how to sort of combat that with environmentally sustainable alternatives. So, yeah.
Does anyone else have a question they'd like to ask?
I was wondering, a lot of data collection in these assignments, that you had any difficulty gathering data from various sources for the people willing and able to share their information with you as a research project?
I think everyone faced their own issues with data collection, but I was lucky enough to be able to consult with 2 different scientists and experts in the area, so I was able to set up Zoom meetings with them, and they were kind enough to share a lot of their data with me and explain a lot of their articles that they had written. So I really enjoyed doing that, but I think, well, for me it was secondary data, so I was able to sort of utilise Excel skills that Dr. Wood and Ms. Woodward had shared with us. So yeah, that's how I collected my data.
Thank you so much for the question. And yeah, kind of similar to Grace, because my topic was like kind of niche in a way of like Haemochus contortus. I couldn't find much. And then obviously I was also researching into a certain vaccine. So it wasn't even like I was looking at many different vaccines, it was a certain one, so I did struggle to find articles that kind of helped me in that way. And then I didn't find ones that were just based on sheep as well. I had to look into sheep and goats and kind of the whole of the agriculture industry.
Yeah, I think everybody who did secondary data, definitely. I think because we all picked very niche projects. So I think everybody struggled a bit, but mine weren't that hard to find. I had like lots of different data sources, data sets that our teachers gave us, so I was able to find one pretty easily. But yeah.
Any last question for this group? I'll do one more from me then. So, Nicola, who do you think will benefit the most from what you've discovered in your project and your results?
Probably farmers, people in the agriculture industry, livestock especially. Well, mine was about prime diseases in general, but I specifically looked at CWD, which is chronic wasting disorder, which is a prime disease, like a zombie disease, that affects cervids, so like deer and sheep and stuff like that. And it's mainly in North America, and so it's affecting the agricultural industry a lot up there. So I think figuring out how to stop it would be good.
I guess Lucy, have you shared your results with your dad, and what did he say?
I have shared them with my dad, and whilst it's not obviously on our farm, like he still found it very like interesting and everything, because it's more found in Western Australia, in like hot, humid areas, not so much at Canamble where I live, but still very, very interesting, like obviously because we do have livestock such as sheep, so it was interesting to learn about areas that other places in Australia kind of like struggle with.
Great. So you had the opportunity to speak to experts in the field. What did you learn from being able to talk to scientists that was more advantageous than necessarily reading their journal articles?
Yeah, definitely overall just in-depth knowledge into the whole topic. Definitely a lot of the issues they faced in terms of gaining research in the field and a lot of the variables that you aren't able to control. Definitely that, and things that weren't mentioned in their articles in terms of resistance that they've noticed, growing resistance to biopesticides, which is now an issue in terms of Helicobacter moths. And yeah, there's also just going in depth into the issue of maintaining refugee crops, which try to prevent the growing resistance. So definitely just kind of overall learning about how they faced issues and also just in-depth knowledge in general, yeah.
Nice. I'd like you to help me. Thank you. Introduce our next group of topics. Which relate to health and sport.
My project investigated diagnostic methods used to identify abnormal brain structures. These are also known as malformations in cortical development, or MCDs. Recent studies have identified lack of conclusiveness in neuroimaging in diagnosing MCDs. Thus, I inquired into the effectiveness of invasive and non-invasive genetic sequencing methods in producing a diagnosis of a particular MCD. Because this area of research was on humans, ethical principles restricted access to primary data. To work around this, I conducted a review of relevant published literature. Data available suggests that sequencing of tissue obtained through invasive diagnostic methods provides a much more accurate method of identifying MCDs due to its high diagnostic rate.
So my topic is investigating the effectiveness of electrostimulation versus natural massage on different muscles with athletes. When conducting this investigation from secondhand data, I found that electrostimulation was more effective in the rate of recovery for these muscles. I tested muscles of the hamstrings and the quads with different exercises such as walking, running and jogging. I found this investigation really important as I thought it was really interesting, and I got to understand what type of rehabilitation method was most effective.
My project is about concussion and the role recovery has in preventing long-term brain damage. Concussion is a commonly overlooked issue in society, which is why I have conducted a second-hand investigation into the impact it has on individuals. Chronic traumatic encephalopathy, known as CTE, is a degenerative brain disorder linked to repeated head injuries, killing the brain cells over time, leading to symptoms such as depression, memory loss, and dementia. This condition cannot be tested for during a lifetime, as it must be found through an autopsy, which shows the importance of preventing head injuries, as this is the only prevention method. A study to assess problem-solving skills and other factors affecting the brain was done by examining mice 2, 7, and 14 days after a concussion. All tests showed that 14 days had a better result than 2 days, supported by a study in the AFL of players returning 0 weeks after a concussion and 2 weeks after, with more receiving a second concussion with no recovery time, since they are more susceptible as the previous injury was not properly healed. My results supported the hypothesis that the more recovery time there is, the decreased risk of a second concussion and will show improved skills. These results are important for sport guidelines, as athletes must follow the minimum recovery time for a concussion in order to reduce the symptoms and chance of a second injury to reduce CTE.
With the issue of cocaine dependency on the rise, I chose to study different treatments for cocaine addiction, specifically genome editing and pharmacological strategies. So, specifically after COVID, there was a huge rise in the prevalence of cocaine addiction. It is estimated that about 1.3 million people worldwide struggle with this. In 2020 to 2021 alone, around 884 Australians died due to cocaine use. So my project, obviously I'm going to do first-hand research, so instead I did second-hand research, and in doing this I looked for a selection of different papers in which I calculated effect sizes using the p-values and the subject sample sizes. And then I compared the effect sizes of the different strategies, including the use of drugs like modafinil, which aim to limit the effect of cocaine once it reaches the brain. And then I compared these studies that use drugs such as that with genome editing, specifically the editing of the BCAG enzyme. And what these studies tried to do was break down cocaine before it reached the brain and before it produced that high. And it was found that the genome editing was far more successful in limiting cocaine-dependent behaviours than the pharmacological strategies were, which matched my hypothesis.
So, for brand new projects, would anyone like to ask these girls some questions about their topics and their results? I might kick it off then. Amy, what did you find surprising about your results, if anything?
Well, I didn't really find things surprising per se, but I think it was interesting in how both drugs did different things. So whilst the results weren't necessarily surprising, because BCAG looks at breaking down cocaine before it reaches the brain, before it can block dopamine receptors and cause a buildup of dopamine, whereas modafinil and similar drugs like that look at limiting the cocaine dependency behaviours after the drugs have reached the brain. So in that way, the result we kind of expected that not having cocaine reach the brain at all would be more effective.
Let's go the other way to Ellie. Why this topic?
So growing up, I played a lot of sports and I'm quite accident prone. So let's just say there's been a few injuries over the years, and especially with my lower back, I've had a lot of massage done over the years, and I think electrostimulation was really just something that I didn't completely understand. And so that's how I formulated my hypothesis in thinking that electrostimulation is going to be more effective for the recovery rate of muscle injuries, which I did find was correct in the end.
So follow up. Not that I wish you to be injured, but should you be injured in the future, what treatment do you think you would ask your doctor about then, based on your project?
From my project, obviously I would ask about electrostimulation. However, it is not a method that is used as often as massage or other physio methods. So maybe I'll just have to stick to my normal massage.
Yeah. Why isn't it used as often? Is there a particular reason, like cost or?
Oh yes, I think it's not heavily accessed as other techniques, because electrostimulation is the processing where like nerve signals in muscles are targeted and to create better impact for the athlete and to affect the muscles faster, therefore recovery rate and recovery time is quicker. But overall, I just think it's an access issue. I wouldn't say cost, maybe. Yeah.
Nice. All right. Well, next up, June. You were lucky to also work with a mentor. What did you learn from working with her?
So I worked with Professor Sandra Cooper from Sydney Uni, and she really helped me just round down my topic. So
So my dad is the clinical lead for Dixie Medical, which is an electrode company. So I just knew that I really wanted to do something surrounding epilepsy or just neuroscience in general. And her area was looking at genomics and particularly diagnosing epilepsy and malformations of cortical development based on genetic mutations. So that was really interesting in looking at her work in sequencing DNA from electrodes that were used in surgery. I just thought that was really fascinating. So just being involved with her was very helpful in rounding down my topic, and she also helped me to gain some data sets, which was very helpful because it's obviously not something that I can really gain primary data on myself. But yeah, that was very helpful.
Does anyone have any questions for this group?
I have a question for Amy, actually. You talked about with BCAGs and stop the receptors in the brain getting the potential benefit of cocaine. How do they implement that in somebody, and why don't they do that before to stop people enjoying bad drugs?
So genome editing as a whole, there are huge issues with access surrounding cost. Using things like CRISPR is generally very expensive, and that just makes it very inaccessible to people, especially in rural communities. In addition, there's not too much research on the long-term effects of it. So we know that short-term it can have some beneficial effects, but these are limited over time. And what was the other part of your question? Sorry.
And why don't they implement it more for people? I think you're expressing it.
Yeah. So yeah, that's pretty much it.
And our other question.
I've also spent quite a few terms at school for this on physio over the years. What is it actually pulses?
Electrostimulation is specifically referring to pulses that target musculoskeletal injuries. Yeah. So, yes, that can be used, yes. I researched that briefly into my investigation, yeah. So it does involve that.
All right, will you please join me in All right, up next we have our next group of projects. And what was their theme? We'll find out. Their theme was global issues.
Hi there, I'm Elise. My science extension involved seeing the difference between orthogonal convergence and the rate of carbon and sediment thickness at subduction zones. My project was really interesting, and the results that I found from it was that carbon and sediment thickness produces higher amounts of CO2 at subduction zones compared to orthogonal convergence.
I looked at volcanoes and their impact on changing global temperatures. Volcanic activity increases each year. The temperature anomaly will also increase. Lots of people live near volcanoes, and it happens in a lot of places, and it's a very relevant disaster.
As someone who has always been very passionate about the welfare of animals, doing my science extension project on reducing the impact of invasive species seemed like a natural choice. I investigated the effect of keelback snakes in mitigating the population of cane toads, and seeing if increasing the population of snakes using specialized breeding program could potentially decrease the population of toads. Since their introduction, cane toads have caused the endangerment and extinction of several native species and becoming an increasing issue. Although a humane way of lessening the population needs to be discovered, as the current way of reducing them is not working. I hypothesized that increasing the number of keelback snakes would decrease the population of cane toads, and results from data sheets and simulations prove this could very well be a solution to the cane toad epidemic.
Hi, I'm Abby, and in my research I looked into the relationship between temperatures and factors that are indicative of climate change, such as pH, salinity, CO2,
found that the data that I used wasn't significant enough to use to conclude like long-term impacts of temperatures on climate change and salinity in the ocean. But I was able to see a real trend in the different reefs that I looked into, like as temperatures increase, the CO2 concentration decreased, and I would have wished that there were longer entries in the data. This is important to me because I've always had a real interest in marine biology, and it's a real world issue at the moment. Like, as temperatures increase, we've seen like everything is changing around us, and I believe my research or further research into this topic will be really helpful in the future.
My question I chose to investigate was how do green spaces in urban environments affect livability factors such as temperature and humidity. I chose this as information surrounding this topic is relevant in our current environment, as increasing populations around Australia have resulted in increased amounts of built areas and therefore decreased amounts of green areas. This creates a demand for information on the impact this may have on the livability of the area. I placed a small green wall into the school and took data on the temperature and humidity levels of the area with the greenery and without to compare them, and then find the impact of the green wall. My hypothesis was that there would be increases in humidity levels and decreases in temperature in areas with the presence of greenery, providing positive impacts on the livability of the area. The results of my investigation found that statistically there was no significant difference between the area with greenery and without. However, the green wall was very small scale and there were numerous limitations to my research, such as the short time frame was installed for, and therefore how young the plants were, and the impact of weather on the area. Taking this into consideration and looking at information from my background research, it all points to greenery having significant impact on humidity and temperature, so we can deduce that greenery does in fact have an impact on the livability of an area.
Does anyone have any questions yet? Oh,
I just have snakes. Yeah. So, are there any predators for those snakes, or will they even come from themselves?
Well, the snakes are a native species to Australia. They're non-venomous, and they do have, they're very small, so birds and other predators can still eat them. So one of the aspects of my project was making sure that by increasing their population, we wouldn't then be starting another epidemic. So obviously I couldn't gather first-hand data because it would be very difficult for me to go to Queensland and do all of that. But looking at my data so far, it appears as though there wouldn't be any issues with increasing their population.
Actually, another question for me, if that's all right. I have got some questions, so that So, I remember reading a post that the killback snake is 70% immune to the poison of the cane toad. Is it possible that they can gain immunity or resilience to the cane toad venom and be more successful?
Yeah, well, the exposure is obviously by process of natural selection. The more exposure they receive, the ones with less resistance to the toxins will unfortunately pass away. But that means the ones who are more resistant will then go on to reproduce and increase the resistance of the snakes. It also means that other species, other native species, eventually might also develop resistance to the toxins, which could also help mitigate the cane toads. It might just take a while.
Although this time, on the green spaces, I thought that was really interesting, by the way, and I was wondering whether you had a chance to look at the mental health impacts and whether it actually just made people happier to have green spaces around them.
Yeah, so I did conduct research into that, and unfortunately in the time I had there was no chance to do any research firsthand into that, but I did find through quite a lot of investigations that there were results saying they increased things like productivity, they reduced crime rates, yeah, they were great for mental health overall, so yeah, they had a lot of benefits to those
might stick with you, Igor, you've got the microphone. Okay. You were one of the few people in this year's cohort who did a primary investigation, and you did it within the school environment. So what was one of the biggest challenges you found actually performing your experiment in the school environment?
Yes, there were a few difficulties, and I think especially to do with weather, which I obviously can't really control. Weather did affect my results a bit, and also, say, students were sitting around the green wall and I can't monitor the green wall all the time, so there's no way of knowing if they might have moved things or touched things which could have affected it. So yeah, there were a few factors just from having it within the school that could have affected my data.
Nice. Miss Abby, you went on the STEM tour, so can you tell us about how the STEM tour impacted your project and the connections you made on the STEM tour fed into your project?
Yeah, so last year on the STEM tour I got the opportunity to work with 3 university professors from Sydney Uni and from the University of Wollongong, along with a student doing his PhD in, I can't remember what it was ocean ridges, which are like systems that work on the borders of reefs and like changing the impacts of waves on like the inside of reefs. It was really interesting, like being able to work with them, and through that I got a lot of advice on like where I can study in the future. And in particular, I reached out to Professor McGregor, who works at Wollongong, and I changed my project like the start of this term, and originally, so it's a bit of a rush. But originally I used some of her data that she collected, so I originally looked at phosphorus levels in corals, and she provided me data that she had collected herself, and gave me advice on how I can use that and where this can take me in the future. But unfortunately, due to other issues with my data, I couldn't find any years that matched up with the data that she gave me, so I couldn't really take it any further. But it really gave me a good insight into possible areas that I could change to, and I didn't actually have to change my project 360 or anything. I could stay on the same track, I just had to adjust a little bit to do it, so it was really helpful.
Nice, thank you. Really, let's go along the road. You actually did some modelling using a simulator as part of your project. So how did simulation feed into the development of your results?
Well, obviously not being able to go to Queensland, I had to resort to either using data sets, and we found some simulators which aren't directly based off of cane toads and snakes, so that was a bit of a limitation. But I was able to vary the numbers on the simulations in order to get different results, and so that, because the results, I didn't have to do multiple trials or anything because they were based off of a formula, so that was useful, but it was also limiting because it wasn't directly related to the fact, to the snakes and toads, and it also didn't take into account that they had other predators or prey or anything. So, yeah.
Nice, thank you. Caitlin, you did something which is quite normal in scientific research, which was change your topic a few times and morphed along the way a bit. So can you tell us about actually the development of your topic and how it changed over your journey?
Well, that's kind of the good thing about science extension. You can have this big, giant idea at the beginning, and then, it becomes a bit unrealistic, and you go, oh, we've only got this short amount of time. I can't even remember what it started off with. And then, data was a bit of a sore point for me, collecting. It took a while. When I finally got it, I kind of had to change it with the data that I had. So yeah, I think I changed my title about 5 or 6 times, and then I changed my hypothesis, and then I changed my data again. So it's not a straight line when you do a chemistry experiment or something like that. You don't just have a hypothesis and independent. You've got to change it a bit. It's a bit tiring, but it worked out in the end. So
you did a good job at real science. that's how it works in reality. Miss Elise, you had random data. You had some nice big random data sets in your project. So you weren't short of data, but you had to use that to find your findings. So can you tell me a bit more about that?
Yeah, so with the random sampling of the data, it actually ensured that there was like bias that was reduced in terms of making sure that as a whole my study was actually really accurate in the end. And without, sampling that random data, it actually would have been not an accurate task at all. So it's really quite a great thing that Sabine from the University of Sydney, who's a very good earth and environmental professor, came in and actually gave me all that data, but not just the data he had, but also the random data as well. So it was great.
It was great to listen to. He was amazing sitting on all those Zooms. Question down the front.
You mentioned that there was a lot of data that you got. Did you have to do much in terms of data cleansing to make it more palatable to the general audience?
Yes, there was like so much data cleansing involved. It was like over 2,000 like sources of data. So I had to like go to Sabine, constantly emailing, him saying like, how do we change things and what would you like me to do? So it was a long process, but we eventually got down to a really nice, concise data set, and it worked out in the end.
Nice. Any last questions from the audience? Go for it.
Caitlin, what was your favourite part of your project?
Probably the volcanoes. I'm a little bit of an earth nerd, only a little bit. So, when I first was trying to think of my project, I was like, what do I like? Ooh, volcanoes. Just went with that.
Why not? Well, if you'd like to join me in congratulating you. So our next group have topics related to just science extension in general, apparently. Here we go. Some eyeballs.
Hi, I'm Sophie. I decided to look at if there was variation in efficiency between quantum and classical computers, and I decided to do so because I've always been interested in quantum physics and more recently the applications in computing, such as improvements in cryptography and medical science and aerospace. I wanted to be part of proving whether there was really an exponential speed up as the media likes to proclaim. So I have hypothesized that there is a variation between speeds and the number of calculations taken for quantum and classical computers. Using the IBM software and an algorithm called Grover's algorithm, my results show that there was an increase in speed aligning to the quadratic speed up of the theoretical trend. However, I had a lack of control over the noise and temperature fluctuations in the publicly accessible quantum computer, so I found there was latency in the speeds, and I also found that there was only significance in larger scale problems rather than the small sample of 1 to 7 qubits in the computer that I use.
So I looked at how does the hypothesis affect the stride length and stride frequency of bilateral transitive entities at maximum velocity. And I chose to look at this because there were many controversies surrounding para-athletes because it's really hard to measure and compare disabilities. And so there's a lot of questions around the level of competition and if they get an advantage with their prosthetics. So many have been disqualified affecting their career. So I was looking at if the height provides an advantage by increasing the speed. And so my hypothesis was that the height of the prosthetic would increase step length and step frequency when at maximum velocity, but this would not affect the maximum velocity. And so the results show that an increase in step length and a decrease in step frequency occurred from the height increasing, which did lead to a constant velocity as they cancel out. So from just the height of the prosthetic, you can conclude that there is no advantage with prosthetics. However, there are many other components and parts of the race to look at. With
climate change becoming an increasingly concerning issue worldwide, the demand for more sustainable and renewable energy sources is growing. The rise of renewables across the world is already evident, and when we consider the alternatives on the market for coal, oil, and gas, it is paramount to weigh up both the positive and negative aspects of each alternative before electing the most appropriate energy source. As of 2019, 84.3% of total energy generated worldwide uses some sort of fossil fuel, and whilst a 1.8% decrease since the year 2000 may seem insignificant, it does in fact show a substantial shift towards the UN's net zero goal. For my study, I analysed ongoing operational data collected by BP from both corporately and government-owned generators between 1971 and 2021. My study compared the installed capacity and generation of fossil fuels, nuclear, hydro, solar, and wind against their respective prevalence in their national energy mixes as a measure of energy sources efficiency. Previous studies have found strong links between economic, geographical, and political factors and energy source prevalence. So to minimise the influence of these variables, a range of countries from different continents and a range of GDPs were selected to analyse. This study can be used as evidence to forecast generation and profit of proposed energy infrastructure developments. However, further efficiency and emission research must be conducted. Ultimately, no conclusions regarding the correlation of installed capacity and national prevalence of generation and national prevalence can be made overall. However, it can be concluded that passive energy sources tend to be more efficient in their production and usage.
Hi, I'm Tilly, and for my science extension project this year, I studied the difference between different methods of estimating the body mass of dinosaurs. It's a bit of a niche topic, but I think I came about it. I've always been really into dinosaurs, I guess, and we had a biology depth study, and I really wanted to look at dinosaurs, the part of it, and I realised that's kind of not biology. So, strictly speaking, so I kind of shifted it to looking at the biology of dinosaurs and how we can think about that. That didn't end up working out, and then science extension came along, and I was like, oh my gosh, that would be an amazing topic to study. So I went into that and eventually kind of narrowed it down to looking at body mass. It's really important because it's one of the most significant, most determinant factor of a lot of biological functions with dinosaurs, such as like metabolism, locomotion, reproduction, all that really cool stuff. Obviously we can't directly look at the biology of dinosaurs because all their soft tissue is decayed over millions of years they've been around. And so this is a really good way of being able to have a bit more insight into all of that stuff, and we can see how we evolved, how things worked, the different climate that was around at the time, all of that stuff. So what I did, I got data from this paper that I looked at volumetric hull, using volumetric hulls, which are reconstructions of a really complete specimen. They're digitized, and then you find the volume, and then you multiply it by whatever constant to get the mass and the density and stuff, and then scaling equations. And those use the relationship between body mass and proximal limb circumference. So that's like the humerus and the femur of these dinosaurs to find the body mass. And what this paper did, they got each of them and they looked at them separately. And I wanted to compare them. So I did. I looked at what the outliers were in each of them. I looked at the similarity, the differences, and then I did a number of t-tests and stuff to find those differences between the mass values that they found. And I found that, yay, there was a difference. And overall, volumetric hulls were more accurate, close to the kind of accepted value of 600 kilograms. And yeah.
Rage from quantum computing to dinosaurs for this group. Do you have any questions for this group?
Louise, you mentioned when we were talking outside that certain heights of aesthetic legs are not allowed in the Olympics. And then your video said that you found no difference in that affecting velocity. So do
research around this area, and there are actually many variables that affect athletes, and there's a lot of contention whether different things actually provide advantage or disadvantage. So there's a few studies that look at this, and there's a lot of different results. So I think these results need to be considered among other variables to actually decide if this has an impact. But it's really important to have a look at because it does affect the careers of many athletes. So, yeah.
Any questions?
So I have two questions. One for Silva and one for Larissa. First, did you find any dinosaurs having prosthetics? The second question was for you, Louise. With the substance within the prosthetic, did that make a difference, or was it, is it really standardised, the prosthetics that you can have for competing?
No, so like the prosthetics are often personalised, and there are also many companies that do different shapes and models and stiffness. So there's a lot more variables with the actual prosthetic, as long as like the part of the race and the actual athlete. So there's many more like variables concerning like the stiffness of the prosthetic changes, and the height or the length of the blade or the actual model or the shape. So there are a lot more variables to consider that do affect it as well. So yeah.
Bronte, you said you're using data from, I think it was 1971 to 2021 from government and non-government sources. So was there a lot of data there that you had to sort through, and if so, how did you condense it down?
Yeah, I actually would go on and put money on the fact that I reckon I have the most data in the cohort. I originally started off with 3 data sets for every energy type, for every single country in the world from 1971 to 2021. And I managed to get that down to 3 data sets for 5 different energy sources for every country in the world between 2000 and 2021, and that was a lot of hours of data cleansing. So, yeah.
You did a good job on that.
Are there any AI tools that could help you cleanse that data more efficiently?
If you know some, let me know. It's a bit late, but it would have been great 6 months ago.
I'll write that down for next year.
Yes, do that. If they can do statistical tests too, that'd be great.
That won't help you in your exam. You used to have to do it. So, Tilly, if you had more time, what else would you do on your topic to expand on the answers that you found? What would the next steps be?
Great question. Well, I do my study off of all secondary data. It's a little bit hard to get access to, fossils and stuff. But I probably would have conducted more first-hand investigation. Probably using like a chicken or something, a little smaller scale. Sorry. And I probably would have tried the different methods in it and used them on that specimen. So I would have had to, it would have been very time consuming. I would have had to learn how to make volumetric hulls, use all the tools and the technology to do that, and then I'd have to find an appropriate equation, or I could not derive it, that is so complicated, but something like that, and I would have compared them myself through that method, which would have been really cool, but maybe in the future, we'll see.
Nice. Projects for the next cohort. Yeah. Bronte, what, if anything, did you find surprising in your results?
I found it really surprising that the results for the energy sources that we use most commonly, like fossil fuels, are really inconsistent in their efficiency. So some of them are just really poor efficiency. And then some is just absolutely zero correlation. And I was doing some more research into the actual relevance and accuracy of my variables in calculating efficiency, because obviously efficiency is usually calculated as just useful energy over produced energy, as a decimal, but I actually did it as the installed capacity and generation as a factor of the national prevalence. So I found that it was interesting that my equation showed a lot more variation in like the actual efficiency of energy sources that we use, and that renewable passive energy sources like solar and wind tended to be the most consistent and most statistically significant. So that was surprising.
Louise, I believe you had the chance to talk to the head of the Paralympics at the very start of your project. How did that help shape the direction of your project?
So I got in contact with a lot of mentors at the AIS, which was really useful because this topic was quite broad, and there's so many possibilities when it comes to prosthetics and sprinting. So it was really helpful just to get a broad understanding of how how prosthetics sprinting is different to normal sprinting, and the differences, but also understanding how prosthetics work. But it was also good because I could start to ask for some data or some ideas and experiments. So it was really helpful just to have those mentors, and also to network if I continue this after school. So it's really nice to have that network.
Nice. Any last questions for the audience? All right, join me in congratulating you. We're on to our last group of projects now, on the theme of health again.
Hi, I'm Georgia. My topic is essentially looking at firmicutes and bacteroidetes, which are two phyla or families of bacteria in the gut microbiome and how they change when humans consume Mediterranean diets versus Western diets. We're finding more and more about how we react to medicine, food, exercise, and how we get different diseases and neurodegenerative diseases, and that it's very relative to how our gut microbiome is composed. I found that whilst the ratio stays the same between these firmicutes and bacteroidetes, interestingly, different species do different things and they're in different abundance. They don't quite follow this ratio. I believe that further research is required before we can really rely on these microbes' changes to make comments on people's health.
My purpose for this project is to provide information that raises awareness around the implications of climate change and the detrimental effects human impact has on contributing to climate change. This study hopes to provide data and information surrounding climate change in the hopes that further action or consciousness can be taken in regard to preserving the natural environment. My topic concerns climate change, which is significantly relevant in our society as there has been a drastic shift towards environmental concerns through social action such as protests and policies. This, as a result, stresses the importance of environmental and climate conservation in society in order to preserve the natural environments for generations yet to come. My hypothesis was that there is an effect of ocean acidification decreasing the population of phytoplankton, which as a result will affect their role in decreasing effects of climate change. My results showed that as pH increases, generally the average number of phytoplankton decreases, supporting my hypothesis. My results also showed the decreasing trend of pH over time, representing an increase in acidity over time. My results also showed that as pH increases, generally the number of phytoplankton decreases. Also, my results showed that as pH increases, carbon increases as phytoplankton become less efficient in removing excess carbon, increasing carbonate. Also, as pH increases, temperature increases, which supports the acceleration of climate change, and therefore the direct relationship between phytoplankton and climate change.
Around 40% of diseases are sex-linked, and over 18 million people are diagnosed with cancer every year, and this number only looks to increase. With over 533 inherited diseases, variations to the X chromosome challenge our understanding of genetic mutations, making the process of tracking and solving various cancers near impossible. However, studies have been conducted with Drosophila spies, displaying the movement of and inheritance patterns of disease to help develop treatment and potential causes. As part of my exploration, I conducted my own primary experiment using white-eyed and amber-eyed Drosophilus flies, breeding them through 3 generations to see how dominant traits would mimic cancer as a dominant gene. Throughout my experiment, I found that diseases are more likely to overpopulate healthy cells, with the white-eyed flies being more prevalent than the red-eyed flies in the end. In relation to my research question, can we track movement of cancer by looking at inter-inheritance patterns of Drosophilus flies, it can be concluded that we cannot rely on inheritance to predict every type of cancer. This is also supported by a research report conducted by Meshev, who found that environmental factors like diet, lifestyle, and bacteria all impact the continuity and prosperity of disease. To improve my experiment, I would have continued to generate the flies for a few more generations and repeat the experiment to improve reliability.
So I studied the correlation between meditative practice and increased grey matter in the brain, and this was due to the neuroplasticity of the brain, which is its ability to permanently change. So grey matter is the outermost layer of the brain and is hypothesized to increase with meditation. So I chose 5 brain areas that were most likely to increase with meditation, based on the brain areas that are involved during meditative practice. So in my results, I found that all areas increased of significance in comparison with non-meditators and meditators, which does suggest a correlational relationship, but further research is required, ideally longitudinal research over long periods of time with long-term periods of meditative practice in comparison to someone who is meditation naive. The complications of the study remained mainly revolved around the confounding variables of the brain, such as gender and diet and nutrition, and which all impact the grey matter in the brain, and so it's quite complex to remove those variables in order to find grey matter on its own. This data and study is of relevance because it has strong links to well-being outcomes, such that meditation increases well-being. And so that helps with the rising rates of mental illness in today's society.
Has anyone saved their best, trickiest questions for last? Amy.
Amber, you said that you were looking at 5 parts of the brain that were most affected in terms of grey matter. Which were these 5 parts of the brain, and why were they more affected by meditation than other parts of the brain?
Oh, good question. Yeah, it was actually quite a complex process to pick the brain areas of interest, because most of the studies just looked at like 30 brain areas, and while I would love to look at all of them, I couldn't really. So yeah, I went through multiple studies and tried to gather knowledge of what their conclusions found. So mainly the brain areas were in the right hemisphere, and that's usually, we hypothesized because meditation is, well, the right side of the brain is usually involved in creativity and like intuition, and meditation kind of triggers those areas in the brain. So we found, well, I didn't, but the studies found that areas involved in those brain areas that were activated during meditation were more likely to be increased, mainly in the temporal lobe. So yeah.
I have to ask my sister, but you talk a lot about like productivity and well-being. What would your recommendations be to employers and also to schools?
Thank you for giving me an easy question.
You stole mine.
I was talking about this with some teachers earlier, but kind of a sub-area of my study was looking at well-being, especially, as I said, with the rising rates of mental illness and disorders in today's society. I would absolutely encourage a well-being or meditation program. I think it's really important. I know some people don't love it, but if you really engage in the benefits of meditation and you acknowledge how much it helps you personally develop and also how you react positively to others, I think it's a really important aspect to introduce if we can, and especially in the working environment as well. It just I ended up actually trying out meditation because of my research, and I think it would just really aid like the cohesion of a workplace environment, and I think it'd be a really good thing to try out. Yeah.
So following on with that, Amber, do you think do you think the results from your study would help convince more people to take meditation?
Oh, absolutely. In one of my background research, mainly not looking at grey matter specifically, but I found some really, really engaging stories of people who had insane outcomes out of meditation. There was one I looked at where this woman had an absolutely awful course of her life. She had developed cancer, her husband had died, and she lost her job. It just had all gone downhill. And she ended up trying some intense meditation methods that changed her brainwaves. And that ended up, obviously we don't know for sure, but she ended up reducing the size of the cancers in her lungs, I believe. Sorry, I've got a bit sidetracked. But yes, I believe it's an insanely helpful thing that we just don't know enough about right now, so it definitely should be encouraged and looked into.
Did you look at the difference between studies in clinical practice and broader population? Can you say the non-clinical settings versus clinical settings in your studies?
Yeah, so that relates to one of the main complications of my study, which was it's a very, I want to say niche because it seems to be a topic of the night, but it is very under-researched. So a lot of the studies that I looked at got participants from Buddhist temples, and because they have a lot of experience in the area, and so would most likely have more significant results. But yeah, some of the studies I looked at took participants just from the random populations surrounding where the study was conducted. But in doing that, they had to minimize like race, gender, diet, all those other factors which would influence that. So there was some complications in that area. But the study I mainly got data from did use people who practice Sahaja yoga meditation in a select environment. Yeah.
Georgia. So you looked into the gut microbiome, quite a complex topic. What would you say are the next steps that need to be investigated to better understand the role of the microbiome in health?
Yeah, okay, so the gut microbiome, unlike Amber's topic, is there's an awful lot of stuff out there. But the thing is, it seems like everyone disagrees with each other, and whenever I end up with like 3 days before the assessment task is due with finding this other paper that someone else says we absolutely disagree with everything that's ever out there. And I just ignore that, we won't talk about that. But because of all these disagreements and because there is so much that we don't know about yet, actually just having something that we then talk about in terms of health and implementing it becomes really difficult when we're still back trying to figure out what is going on and like how to use it. And so if I was to say like hypothesize how this would turn out and implement in the health community and use it, it would become some massive like human genome project size thing where you would take in 3 stages, you've got to figure out what a microbe in a lot smaller detail than what I did, because I did really big like one step down for bacteria rather than like individual species. You need to do individual species and look at what does that microbe do, what metabolites does it create, what do these metabolites do then, and how do they interact with other metabolites and other bacteria around. And then you need to look at how do you increase that or decrease it, depending on whether you want that metabolite or not. And then also figure out, then once you've got all these metabolites, how do you personalise it for different people, because you wouldn't suddenly be like, butyrate is a metabolite that's produced by a certain bacteria when you eat an awful lot of pomegranate. I don't know why that came up, but it did. Yeah, so that is in the Mediterranean diet a lot more. But you take something like that and consider that you want this metabolite, but you've got other bacteria already present in the body, and so would you be able to just straight up say eat more pomegranate, or would you have to say you have enough of this other one to support the system of that one, that bacteria? You can't just immediately say eat pomegranate, you've got to be like eat these other things too, and it all come into ratios, and you have to generally advise people until they do these massive studies in advance. If you eat too many firmicutes, which is one of mine, then you end up with obesity is very large with people with high firmicute numbers, and then bacteroidetes. If they eat too much of that, then you can get irritable bowel syndrome, and that's what the Mediterranean diet is on the way towards. So balance is better. As we know throughout most of diet stuff, everyone's like, eat a healthy, balanced diet, and it comes back to that.
So, follow-up question to that one. Have you just mapped out your career pathway?
Should I be running a new human genome project and make it an international corporation thing? A 50-year project or whatever that was.
So obviously all different kind of gut bacteria and all that can be very individual. Did you struggle to find data that was comparable and relatable in the fact that they looked at, due to the fact that they must have looked at different microbes and such?
Yeah, so what I actually originally started with just generally, is there a really broad bad idea, of is there a difference between, I'm going to start with firmicutes and bacteroidetes. I had, is there a difference in the bacteria they produce, and so I just looked at a random group of, they all do fecal samples is how they get this data, by the way, and so they test it and have a look at, come up with this huge list of different bacteria at all different stages. So some studies looked at species and others looked at genus levels and stuff, and it made it really difficult because a lot of time they didn't cross over, which is what I ended up doing with and checking from firmicutes instead. And so I ended up doing all firmicutes, and any information I found on any bacteria, I just Googled it and was like, is this a firmicute, is it bacteroides, or is it one of the other 4 subgroups in bacteria that's in the gut microbiome? And for all the other ones, I ignored them because they're not as prevalent, but then I put them under the Firmicutes-Bacteroides, and that was my way of kind of grouping them so I didn't have to deal with the crossover, because things like Faecalibacterium prunense is a genus level 1, and then you end up with others that are, it's a type of firmicute, but then you end up with other ones that are much like larger scale groups of bacteria, and yeah, they kind of don't cross over. So that was how I sort of dealt with that. Just an awful lot of figuring out which categories some things were in, and that's why you end up with the differences in the ratio. Overall, people were like, firmicutes much higher in the Western diet, but when you look under firmicutes, almost like half of them, or more than half of them, the actual individual species are higher in the Mediterranean diet anyway, which is why I would say so much more needs to get done, because you can't just suddenly say, eat things that make you have firmicutes, because it does the opposite in like certain ones. Certain species do different things, so it's kind of doesn't work.
Nice, thank you. Miss Charlotte. Obviously, climate change is a very prevalent and important topic of today's society, but can you tell us a little bit more about the role of phytoplankton in this big picture story, and why phytoplankton is important to talk about?
So I think that there's an increased awareness of climate change. So I found that as pH increases, phytoplankton's population decreased as the conditions were less favourable. And phytoplankton play a significant role in the carbon cycle by absorbing excess carbon in the atmosphere, and then when they pass away, they sink to the ocean floor and they take their carbonate shells with them, and that's basically how they remove carbon from the atmosphere. So by pH increasing due to climate change and increasing acidity in the oceans, it has a significant role on their population, and therefore their role in absorbing the excess carbon. So yeah, I just found that over time acidity was increasing, and temperatures as well due to climate change, and just stressing the importance of environmental conservation and animal conservation, because they play such a significant role in our environment. So I think it's really important that we start to prioritise that conservation.
So that is the end of all of our groups. So I'd like to actually invite all of the 2023 Science Extension cohort up to the stage so we can celebrate them all together as a group cohort.
That brings us to the end of our evening. Both Dr. Wood and I have had the honour of working with these students. And I say that because they've taught us a lot. I think the only thing I can say I've taught them is how to use Excel. Some of them love it now. Lucy?
No, we'll never agree on it. We never want to see another Excel spreadsheet again.
You love it, you know. They have, and as we keep telling them, they are the experts in this. They taught us so much. Every year I say to them, I'm learning more all the time. Well done, and congratulations again. And on that note, we'd like to invite you out, if you have not had a chance to look at their posters and talk to the girls about their wonderful work, please do so before you leave.
Super quickly, sorry, don't mean to interrupt you, Mr. Wood, but just on behalf of all of the Science Extension girls, we'd honestly just like to thank Mr. Wood and Dr. Wood. There's no way we could have done all these so different projects, like on so many different spectrums without you guys. You taught me how to use Excel, didn't quite enjoy that. But no, you guys have done so much with helping us with data sets, and words cannot describe how much you've done for us and the support you've given us. So big round of applause.
certainly has she just sick today. She stated I mean I hope she can't.
She's not.